Look at a single neuron, right now, deep in your brainstem, in a cluster of cells about the size of a grain of rice, sitting just behind your ears at the base of your skull. That neuron is firing. It releases a small molecule called norepinephrine into the rest of your brain about once every 2 seconds. That firing is the reason you are reading these words. In 16 hours, when you are lying in bed and the lights are out, that same neuron will go quiet. It will stop firing. And in the 4 minutes that follow, your body will become a different machine entirely. I want to tell you what happens in those 4 minutes. I want to tell you because I have spent a fair bit of my own life curious about consciousness, what it is, how it switches off and on, why a third of our lives is spent in this strange state we call sleep. Once I climbed into a sensory deprivation tank in California and spent hours floating in salt water in pitch dark, trying to catch the moment my mind let go of the world. I never quite caught it. The biologists, working with electrodes and chemistry rather than introspection, have caught it. And what they have figured out, by listening to electrical activity at the scalp and watching molecules diffuse through brain tissue, is that the engineering inside your own head is not what you think it is. It is not a switch. It is a cascade. And once you see the cascade, you understand why a sleeping pill does not actually replicate it. Now look, the brain is not one thing. When people say the brain, they are doing what physicists do when they say the atom. It is a useful word. It hides everything interesting. There are 4 structures you need to know about for what I am going to explain. Find them in your head. They are in there right now. First, the locus coeruleus, the cluster I just mentioned. About 15,000 neurons on each side. Behind your ears, base of your skull. Pencil-shaped, pigmented blue, which is where its name comes from, the Latin for blue place. This is the wakefulness pump. It pumps norepinephrine. Second, the suprachiasmatic nucleus. Behind your eyes, just above where the 2 optic nerves cross. About 20,000 neurons. This is your clock. Real, physical, ticking. A piece of biological machinery synchronized to sunlight through a direct fiber from the retina. If you removed it and put it in a dish, it would keep ticking on its own for weeks. Third, the ventrolateral preoptic nucleus. A small cluster in the front of the hypothalamus, sitting just above the roof of your mouth. About 4,000 cells. The off switch. Fourth, the thalamus. Egg-shaped, about the size of a walnut, sitting in the dead center of your head, equidistant from your ears. Every signal from your senses, sight, sound, touch, taste, passes through the thalamus before reaching the cortex. It is the relay station. Without it, the cortex is deaf and blind. These 4 structures, talking to each other through chemistry, molecules diffusing across synapses, ions flowing through channels, electrical signals moving at 300 meters per second along myelinated axons, they run the entire transition from awake to asleep. There is no magic here. There is no mystery. There is physics, and there is chemistry, and the rest is engineering. Let me show you the first transition. You climb into bed. You close your eyes. Now what? The suprachiasmatic nucleus, the clock behind your eyes, has been monitoring the light hitting your retina all day. As the light dropped, sunset, dim room, screen off, it began signaling the pineal gland, a tiny structure deep in the brain, shaped like a pine cone, to release melatonin. Melatonin is a small molecule, derived from the amino acid tryptophan. Its concentration in your blood rises about 10-fold from afternoon to midnight. Now I have to be careful here, because melatonin is not what most people think it is. Melatonin does not put you to sleep. The bottle in your medicine cabinet is mislabeled in your imagination. Melatonin is a signal. It is the molecule by which the clock tells the rest of the brain that it is night. The actual sleep machinery is downstream. While melatonin is rising, another molecule is also rising. Adenosine. This one is a byproduct of energy use. Every time a neuron fires, it burns a molecule called adenosine triphosphate, the energy currency of the cell, and a piece of that molecule, the adenosine, is left over. After 16 hours of being awake, your brain is swimming in adenosine. Caffeine, by the way, blocks the receptors for adenosine. That is its only trick. It does not give you energy. It hides the molecule that says you have run out. So now, lying in bed with your eyes closed, you have rising melatonin, high adenosine, and a third actor I have not mentioned yet, orexin. This is a peptide produced by about 70,000 neurons buried in the lateral hypothalamus. 70,000. In a brain of 86 billion, a tiny population of cells whose only job is to keep you awake. Masashi Yanagisawa, working at the University of Texas Southwestern, discovered them in 1998. He also discovered almost immediately that the disease narcolepsy is caused by the autoimmune destruction of these neurons. People with narcolepsy have lost their orexin cells. Without those 70,000 neurons firing, no amount of willpower can keep them upright. Tonight, those 70,000 neurons go quiet. The melatonin rises. The adenosine accumulates. The ventrolateral preoptic nucleus, the off switch, is activated. It begins firing. It releases two inhibitory neurotransmitters, GABA and galanin, onto the locus coeruleus. GABA is the brain's main inhibitor. It hyperpolarizes neurons, makes their interiors more negatively charged, and a more negatively charged neuron is harder to fire. The locus coeruleus, the wakefulness pump, slows. Norepinephrine release drops. And here is where the 4 minutes begin. Across your cortex, a wave of change. The dominant frequency of your brain's electrical activity shifts. While you were awake, your cortex was producing what is called alpha rhythm, about 10 cycles per second, 8 to 13 hertz. As norepinephrine falls, the rhythm slows. Theta waves appear, 4 to 7 hertz. Your eyes begin to roll slowly under closed lids. Hypnagogic imagery, those strange fragmentary images that come just before sleep. The falling sensation, the sudden flash of a face you do not recognize, begins to flicker. Sometimes your leg kicks, the famous hypnic jerk. Almost everyone has had one. That kick is the brainstem motor system briefly reactivating because the inhibitory wave has not yet fully landed. A motor neuron that was supposed to be silenced fires anyway, and the signal travels down the spinal cord, and the muscle contracts, and you twitch awake for a second before falling further down. You are in N1, stage 1 of non-REM sleep, the lightest stage. You can be woken with a whisper. This phase lasts somewhere between 1 and 7 minutes. For most people, 4 minutes during which the wakefulness pump is decelerating, the clock is broadcasting its night signal, the adenosine is binding, and the cortex is reorganizing its rhythm. Then the next transition begins. But the same molecule that is silencing the locus coeruleus is now beginning to act on a different structure entirely. GABA, the inhibitor, is now flooding into the thalamus. Remember the thalamus, the relay station. Every signal from your senses passes through it before reaching the cortex. While you are awake, the thalamus operates in what neuroscientists call tonic mode. Its neurons fire steadily, faithfully passing along sensory information. You hear the refrigerator hum. You feel the sheet on your skin. You smell whatever is in the room. All of it is reaching the cortex through the thalamus. When GABA arrives, the thalamic neurons switch modes. They stop firing tonically. They begin firing in bursts, separated by silences. This is burst mode. The thalamus is no longer relaying. It is gating. And here is something I find wonderful. The bursts are not random. They are rhythmic, 12 to 14 hertz. Each burst about a second long, coming every 3 to 10 seconds. These are sleep spindles. You can see them on an electroencephalogram, a recording of brain electrical activity from the scalp as little oscillating bursts that look exactly like a spindle of yarn. That is where the name comes from. What is the spindle for? It is closing the gates. Each spindle is a coordinated burst that disconnects the cortex from the outside world. The louder the noise outside, the more spindles a sleeping brain produces. People who produce more spindles are harder to wake. This was demonstrated by Jeffrey Ellenbogen, then at Harvard Medical School, working with Robert Stickgold. They played sounds at sleeping subjects and measured both spindle density and the threshold at which the subjects woke up. The relationship is direct. More spindles, deeper isolation. More spindles, longer night. But the spindles are doing something else. They are coordinating with the hippocampus, the seahorse-shaped structure deep in your brain that stores new memories. Every spindle in the cortex is paired with what is called a sharp wave ripple in the hippocampus, a brief high-frequency burst at 150 to 250 Hz. This pairing, this exquisitely timed coupling, is when memories from the day are replayed. The hippocampus chirps. The cortex listens. A piece of yesterday becomes a piece of you. Jan Born at the University of Tübingen has demonstrated in dozens of studies that disrupting spindles disrupts memory consolidation, pharmacologically suppress them, and what you learned that day does not stick. Enhance them, and there are now experimental ways to do this with carefully timed sounds played during sleep, and people remember more. You are now in N2. The bulk of the night, about half of total sleep time, will be spent here. Your body temperature is dropping. Your heart rate has slowed by 10 to 20%. Your blood pressure has dropped 6 to 8%. You can still be woken, but it takes a hand on your shoulder. Then another transition begins. The same gating that quieted the senses is now spreading down through deeper layers of the cortex itself, and as it does, the entire electrical landscape of your brain undergoes a phase transition. The slow waves arrive. A slow wave is exactly what it sounds like. A wave of electrical activity that sweeps across the entire cortex at half a hertz to 2 hertz. That is one wave every half second to 2 seconds. These waves are enormous. They are the largest electrical events in the human brain. They can reach 200 microvolts at the scalp, which is roughly 10 times bigger than what you measure during waking activity. What is a slow wave physically? It is a synchronized oscillation of millions of cortical neurons. They go silent together, a phase neuroscientists call the down state. Then they all fire together, the up state. Down, up, down, up, once every second. A choreographed pulse across the entire roof of your brain. When slow waves dominate, you are in N3. Slow wave sleep. Deep sleep. Delta sleep. Different names, same stage. Roughly 15 to 25% of the night. Now here is what I want you to see, because this is where the night earns its reputation as restorative. In 2012, Maiken and Nedergaard, working at the University of Rochester, demonstrated something we did not know was possible. The brain has its own waste clearance system. She named it the glymphatic system, a portmanteau of glia, the support cells of the nervous system, and lymphatic, the body's other drainage network. What she found is that during slow-wave sleep, the cells of the brain physically shrink. Not by much. About 60% of their interstitial volume opens up. Cerebrospinal fluid, the salty, clear liquid that surrounds the brain, is then pumped through these enlarged channels by pulsations of the arteries. It washes through, picks up metabolic waste, including beta amyloid, the protein that accumulates as plaques in Alzheimer's disease, and carries it out through perivascular drainage into the lymphatic system of the neck. Cerebrospinal fluid moves through your brain during deep sleep at about 10 times the rate it moves during wakefulness. Beta amyloid is cleared at twice the rate. This was published in the journal Science, 2013. The implication is direct. Deep sleep is when the brain takes out the trash. If you do not get enough N3, the trash piles up. This is not a metaphor. This is fluid mechanics. There is a literal river of cerebrospinal fluid pulsing in time with your heartbeat, washing the spaces between your cells. The poetic phrase, letting the day wash over you, turns out to describe an actual nightly hydraulic event in your skull. Picture the person, 68 years old, retired, wakes at 3 in the morning, lies in bed for 2 hours, finally falls back asleep at 5, and wakes at 7 feeling like a wreck. Has been told by the doctor to try a sleeping pill. Has been told by a neighbor to try melatonin. has been told by a magazine to try a weighted blanket, has not been told that the deep sleep machinery in their own head has been declining since age 30, and that the right interventions, morning sunlight on the retina, late-day exercise, a cool bedroom, no alcohol after 8, no screens within an hour of bed, directly target the structures we have just been discussing. People over 65 spend significantly less time in slow-wave sleep than people in their 20s. 20-year-olds spend about 25% of the night in N3. 70-year-olds spend about 5%. The decline begins around age 30 and accelerates after 50. Whether this decline contributes causally to age-related cognitive decline is one of the most active research questions in the field right now. But none of this, the gating, the slow waves, the cerebrospinal flush, has prepared the body for what comes next. About 90 minutes after you fell asleep, the slow waves break. The thalamus depolarizes again. The cortex re-engages. Brain electrical activity, measured at the scalp, looks almost identical to wakefulness. Fast, low amplitude, desynchronized. If a stranger walked in and saw only the brainwave trace, they would say you are awake. You are not awake. You are in REM, rapid eye movement sleep. And here is the strangest engineering choice in the entire sleep cycle. While the cortex is firing at near-waking intensity, the brainstem sends a powerful inhibitory signal down the spinal cord. Two neurotransmitters, glycine and GABA, flood the motor neurons that control your skeletal muscles. The motor neurons hyperpolarize. They cannot fire. Your arms, your legs, your trunk, everything from the neck down except your diaphragm and the muscles of your eyes is paralyzed. This paralysis is called REM atonia. Without it, you would act out your dreams. We know this because there is a disease, REM Sleep Behavior Disorder, in which the inhibitory pathway breaks down. People with this condition kick, punch, leap from bed, sometimes injure themselves or their partners. Carlos Schenck and Mark Mahowald at the Minnesota Regional Sleep Disorders Center characterized this condition in a series of papers beginning in 1986. They also discovered something darker. REM sleep behavior disorder is one of the strongest predictors of Parkinson's disease. About 80% of people who develop it will, within 15 years, be diagnosed with Parkinson's, or a closely related neurological disorder. The brainstem damage that causes the atonia failure also causes the motor symptoms decades later. The disease announces itself in sleep before it announces itself in walking. So during REM, the cortex is awake. The muscles are dead. The eyes move. Why the eyes? Because the inhibitory pathway specifically excludes the cranial nerves that control the muscles of the eyes. The eyes dart back and forth, scanning whatever the dreaming cortex is constructing. What is REM doing? Two things, mainly. It is processing emotion. Matthew Walker at the University of California, Berkeley, in a long series of papers, has shown that REM-rich sleep is necessary for the next-day reduction of emotional reactivity. The amygdala, the brain's threat detector, calms down after a night of REM. People who are REM-deprived, especially through alcohol, which suppresses REM specifically, show exaggerated amygdala responses to negative stimuli the next day. Fear is sharper. Anger is louder. The world is scarier when you have not had enough REM. It is also generating dreams. Allan Hobson at Harvard with Robert McCarley proposed in the 1970s that dreams are the cortex's attempt to make narrative sense of random brainstem signals. The activation synthesis hypothesis. It is still controversial. Other theories, like Mark Solmes's revision, argue that dreams are driven by motivational systems in the midbrain. Both can be partly right. We are not certain. REM lasts about 10 minutes in the first cycle of the night. In the last cycle of the night, just before you wake up, it can last 40 minutes or more. The architecture of sleep is not flat. The first half of the night is dominated by deep, slow-wave sleep, the trash collection, the hydraulic flush. The second half is dominated by REM, the emotional integration, the dreaming. This is why a person who sleeps only 4 hours skips most of their REM. They get the trash collected. They do not get their feelings sorted. They wake up unprocessed. Now let me pause here, because I have told you a lot of things, and you should not believe all of them with the same level of confidence. Let me put what I have said into 4 drawers. Bottom drawer, the things I trust completely. The 4 sleep stages, defined by Eeg patterns, were established in the 1960s by the work of Allan and Anthony Cales. They wrote a manual that every sleep lab still uses. The discovery of REM by Eugene Aserinsky and Nathaniel Kleitman in 1953 at the University of Chicago has been replicated in every species ever studied. The neurochemistry of the sleep-wake switch, orexin GABA, the locus coeruleus, the ventrolateral preoptic nucleus, is settled. You can break the system in predictable ways with predictable results. Bottom drawer. Bedrock. Second drawer. The things that are well supported and recent enough to feel new but solid enough to act on. The glymphatic system, demonstrated by Nedergaard, has been replicated by independent labs. The functional role of sleep spindles in memory consolidation has been shown across dozens of studies in multiple species. The connection between REM atonia loss and future Parkinson's is one of the most robust findings in clinical neurology. Second drawer. Third drawer. The things that are still under active debate. Whether dreams have specific functions, emotional processing, threat simulation, memory integration, is contested. The Hobson activation synthesis model has lost ground to more recent theories. The exact mechanism by which slow-wave sleep clears beta-amyloid in humans as opposed to mice in a laboratory is still being measured. Third drawer. Top shelf. The things that are more inference than measurement. The claim that the decline in deep sleep with age contributes causally to dementia, while plausible and consistent with the mechanism, has not been definitively proven. We see correlation. We have a mechanism. We do not yet have a controlled trial showing that improving deep sleep in older adults reduces dementia risk. That trial will take 20 years to run. Top shelf. Hold these lightly. You may have to put them back. I tell you this because the field is full of people who will not. They will tell you the dream meant something specific. They will tell you the sleep tracker on your wrist measures REM with precision. It does not. Most consumer trackers cannot reliably distinguish REM from light sleep. They will tell you the supplement they sell will increase your slow-wave sleep. Most of these claims belong on the top shelf, but they are being sold from the bottom drawer. You should be skeptical of the people who are too certain. Now let me tell you about Zolpidem. Zolpidem is the molecule sold under the brand name Ambien. It is the most prescribed sleep medication in the United States. Roughly 40 million prescriptions are written each year. The patent expired in 2007, but global sales of Zolpidem and its chemical cousins are still in the billions of dollars annually. Here is what Zolpidem does. It binds to a specific subtype of GABA receptor, the one containing the alpha-1 subunit, and makes that receptor more responsive to GABA. The same GABA we just discussed. The same neurotransmitter your ventrolateral preoptic nucleus uses to silence the locus coeruleus when you fall asleep naturally. Zolpidem makes the brakes work harder. The wakefulness machinery quiets faster. You lose consciousness. But notice what Zolpidem is not doing. It is not entering the suprachiasmatic nucleus. It is not adjusting your circadian clock. It is not increasing the number of sleep spindles. It is not initiating slow waves. It is not coordinating the spindle ripple pairing that consolidates memory. It is not driving the cortical depolarization that produces REM. It is not orchestrating the architecture of the night. It is suppressing wakefulness and leaving the architecture to run as best it can without proper coordination. The result, measured in laboratory studies, is that Zolpidem-induced sleep has reduced slow-wave activity, reduced spindle density, and altered REM. People wake feeling like they slept because they were unconscious for 8 hours, but the restorative work was incomplete. Memory consolidation is impaired. Reaction time the next day is slower than it would be after natural sleep. The clinical literature on this is extensive. Daniel Bicey at the University of Pittsburgh has been writing about it for decades. I want to be precise. Sleep medications are not nothing. For someone in acute insomnia, for the partner of a recently deceased spouse, for someone in a crisis where the absence of sleep itself becomes a danger, a few weeks of pharmacological help can be the difference between functioning and not. The drug exists for a reason. There is a place for it. But chronic use is a different question. Long-term zolpidem use is associated, in observational studies, with cognitive impairment, with falls in the elderly, with motor vehicle accidents the morning after a dose, and with dependency. The drug does not generate the architecture of sleep. It bypasses the architecture of sleep. The architecture is generated every night, for free, by structures in your own head that you have been growing since you were a fetus. The locus coeruleus that quiets, the thalamus that gates, the cortex that slows then speeds, the brainstem that paralyzes the body to protect it from the dreaming mind. 4 structures in conversation. 4 minutes between each stage. 90 minutes per cycle. 4 to 6 cycles per night. A nightly performance of unbelievable precision, generated by a brain that weighs 3 pounds and runs on the energy of a dim light bulb. A pill cannot replicate this. A pill can knock you out. So back to the 4 minutes. You lie down. You close your eyes. The clock behind your eyes has already begun the night's chemistry. The off switch in your hypothalamus begins firing. The wakefulness pump quiets. The 70,000 orexin neurons go silent. The thalamus closes the gates. The cortex slows. The slow waves arrive. The cells shrink. The cerebrospinal fluid begins its pulse through the channels between cells. 90 minutes later, the cortex reignites, and the body goes still, and the eyes begin to dance under their lids. And the cycle repeats, 4 to 6 times, every night, for your whole life. This is not a process you do. This is a process that does itself, if you let it. Your grandmother said, get to bed at a reasonable hour. The locus coeruleus, the suprachiasmatic nucleus, the ventrolateral preoptic nucleus, every one of them is calibrated to a regular schedule. Erratic timing scrambles the chemistry. Your grandmother said, a dark room and a cool one. Melatonin release is suppressed by light, especially blue light, which the melanopsin photoreceptors in your retina are tuned to detect. Slow-wave sleep is enhanced by a cool core temperature, which is why the body actively drops its core by about 1 degree Fahrenheit during the first hour of sleep. Your grandmother said, do not eat or drink late. Adenosine clearance is faster on an empty stomach. Alcohol suppresses REM. Caffeine has a half-life of 5 to 6 hours, which means the cup at 4 in the afternoon is still partially in your bloodstream at 10 at night. Your grandmother said, get out in the morning sun. The suprachiasmatic nucleus is entrained by morning light striking the retina. The earlier and brighter that light, the cleaner the night. She did not know about the locus coeruleus. She did not know about glymphatic flow. She knew about the result. She watched, over the course of her life, what worked. She kept what worked. She passed it down. The science we have built since is in many ways a long-delayed agreement with the woman in the kitchen. You have 4 minutes between each stage. You have 90 minutes per cycle. You have 4 to 6 cycles per night. That is the engineering. Honor it. Lie down. Close your eyes. Let the machine run. Tell me, when did you last wake up, feeling that the night had done its complete work, without a pill, without a tracker, without a tea? Describe that morning to me, because that morning is the experience of the cascade running clean.